Great. So Dr. Charles Edwards was raised in California's San Joaquin Valley, and he worked on his family farms, his family's vineyard near Lodi. He received his Bachelor of Science, Master's, and doctorate degrees in food science from Oregon State University, Cornell, and Penn State, respectively. He's been a professor at Washington State University slash Idaho, University of Idaho in the Food Science College for 32 years. And his focus has been on problem alcoholic fermentations, non-Saccharomyces yeast, malolactic fermentation, spoilage issues like Brett, lactobacillus, and pediococcus. So today he'll focus just on Brett. He's had, as he'll talk to you, he's had a long-standing research project for the Washington wine industry. And just in the last year, he's completed that and he's kind of shifted focus. So we thought this was a good time for him to really wrap up, not just like the last 2 years of the project, but sort of what we learned through all of this. So with that, I am going to turn it away to you, Charlie.
Well, thank you, Melissa. It really is good to be here, gang. I think if there's one positive thing in our world right now, it's been the application of Zoom. And it's just amazing as to our ability now to move information that we didn't have, 10 years ago at all. So it is really good to be a part of this webinar. Just so we're okay, that's me. And the other, actually the good looking one who's behind the tractor, that's my number one son. Well, we are talking about Brett, and I would encourage you to jot down questions and such, because we'll talk about some things that are maybe a little more controversial. Sources, I think most people know that used barrels that are moving around within the industry can certainly serve as a vector, if you will, for Brett. Unfiltered wine. Every once in a while, I'll get someone calling me saying, "What shall I do?" I imported some unfiltered wine from a neighbor, and now I've got Brett. Well, yeah, there's things we can do, but what we should have thought about was filtering before bringing in the wine. Brett has long been thought not to move around by air, but we did some work several years ago now to show that it can. And it's always those situations where you've got an infected wine that you're maybe topping off barrels or moving around wine, and workers turn around and you have Mount Vesuvius coming out of the barrel. So you're getting atomized droplets everywhere. And if that wine was infected, that's an opportunity for Brett to move around. I think all the things that we can talk about, this has got to be one of the more controversial. I can remember when I was a much younger wine microbiologist having arguments at meetings, and all you had to say was, "Well, Brett's never found in the vineyard," and there'll be other wine microbiologists there saying, "Oh, yes, it is," and then the sparks fly. There was an article that appeared in one of the premier wine micro labs at the University of Bordeaux, and she felt that, at least in the analyses that she was seeing and the data that she was examining, that the grapeberry under certain circumstances could be a primary source. Looking through the literature now, I think to summarize most of the recent work, they intermittently will find the yeast on grapes and in must. So it seems to occasionally be there. It depends so much on situations, conditions, things of that nature. But to say it's not found in the vineyard is simply, I'm not sure I'd buy off on that now. This has got to be one of the best quotes I have ever heard about Brett, and I had to put it in. It came from a good friend of mine who has since retired, and I miss her to death because she was, without a doubt, one of the finest wine microbiologists that's ever been around. But she once said that Brettanomyces is nowhere, yet everywhere, all at the same time. And I just think that's an absolute true quote, because you may think you don't have Brett anywhere in your winery, but then all of a sudden, for unknown reasons, you've got an infection, you've got a bloom. So I had to put that into this seminar. So this is going to be a culmination of a lot of different grad students, a lot of different projects. And so we're going to be talking just in general about all these different topics: the role of SO2, survival in pomace, filtration as to what are the needs to get rid of the yeast. Is there any impact of must nutrition on subsequent blooms and infections? What about some of the newer products that are out there, like chitosan? Is that useful? Is it effective? And some of our work on oak barrels I'm going to present as well. And that's work I have to admit, I'm really pretty proud of that. That was a pretty cool project. Okay, SO2. One of the major questions that wine microbiologists have been debating is whether SO2 addition to a wine can somehow induce this physiological type of state. And it's called, let me get my pencil here. It's called VBNC. Very controversial. There are some microbiologists that really disagree with the concept. There are others that are buying off on it. It stands for that. So you believe that the organism is still alive, it still can undergo metabolic processes, but for whatever reason, it will simply not grow or multiply on standard microbiological media. And this could be a real concern, because if it's valid, then the yeast could go undetected using traditional microbiological approaches. So the question begs itself, well, how do you detect this if it's real? Well, there's different methods out there in the literature. You can look at metabolic activity. You can use maybe qPCR. There's questions about the latter, because some are not sure whether you're detecting only alive cells, but you could also be detecting dead cells. We took a different approach. We used something called fluorescence microscopy. We are adding different stains and different dyes that do different things. And they can either tell you that it's alive, as if you get a green fluorescence, or if it's dead, and you get an orange or red. So I think this is a really cool picture. This came out of a wine in which we had just added 0.8 milligram per liter molecular SO2. So there should have been enough SO2 in this wine, literally to kill a horse, if you will. But here you see dead cells. Okay, but you still have these cells that appear viable. And so this started to really pique our interest in this particular concept. So we did a series of experiments in which we plotted population and molecular SO2 against time, using different methods to detect the presence of Brettanomyces. And this just happens to be a strain of Brett that we found in a Washington wine. So here we're adding a lot. We're adding a lot of molecular. There's culturability using a standard plate count type of a medium. And Brett responds exactly how we think it's going to respond. Here you've got a really large population, which I hope no winery will ever have to face. This is over 1 million cells per milliliter. So I hope never to see this commercially. But you can see it instantly went down to levels that we couldn't even detect it, couldn't even see it. But if we look at the culture itself in the wine using fluorescence microscopy, there are the counts of dead cells. And that kind of makes sense. Okay, we started with some level here and it rose over time. That makes sense. But here's the live cells. And that makes sense too, that it declined before it started to increase. But the key is right in here. In one method, you detect them, and in the other, you don't. And here's the decrease in molecular over time. So we think the concept is real. We think SO2 does keep Brettanomyces, their metabolism at bay. And it is one method that can help control, but it does not kill. And that's an important point. Well, one of the questions we also had was about winery waste, because if you've got a winery that has an infection issue, odds are good it's going to be in pomace at some point. And what happens if that pomace is put into the vineyard? So in other words, are there enough nutrients to support the growth of Brettanomyces? And as we've talked about, the presence in vineyards has been long debated. And if all of this is real, that Brett could grow on pomace, how should you treat it before you dispose of it? Can Brett survive the cold winters that we do have here in the Pacific Northwest, in pomace? What about the use of heat? Can that eliminate the organism from pomace? Composting's a harder one to work on, but at least through some temperature data that we've collected, we can at least get some ideas, and I'll come back to that one. So we did an experiment looking at the survival in vineyards, and this took a long time to design, but I have to give some significant credit to the grad student, whose surname was Cartwright, Zach. Wonderful, wonderful researcher. We use filtration devices. We put the pomace in the bottom. This allowed for air to move back and forth. So humidity, buildup of humidity, buildup of atmospheric condition, gas, stuff like that. That wasn't an issue with the experiment, but it kept microorganisms, mold in particular, from getting inside the pomace. So we actually had two pomace types. One that was pressed off when the wine was completely dry, so the amount of sugar would be very small. One in which there was still some residual sugar in that particular ferment. So there'd be some level of fermentable carbohydrate present. We then further split it into an additional experimental design. Whereas for half of the samples, we gamma irradiated them. That way, every single thing in that pomace was dead. We can then inoculate Brett under a pure culture situation to see what would happen. We actually had 3 vineyard sites, 2 in the Columbia Valley AVA, and this was between fall of 2014 to the spring of 2017. And Walla Walla, we had 1. And we collected samples every 10 weeks, and we looked for the recovery of Brett using various microbiological means. All right, here's some of the data. First off, it's interesting that here are the sterilized media, the sterilized pomace. So here we've inoculated Brett into that pomace. And you can see we've got culturability on this side against time this way. So this was a pretty long experiment, 130 weeks. And you had summers about in here, and winters in between. And here's got the 3 vineyards, vineyard 1, 2, and 3 in green, blue, and in red. For the sterilized, you can see we had a pretty good population to start off with. And yeah, there was kind of some up and downs, but they were still culturable way out here by day 130 in sterilized situation. In an unsterilized, we had all different yeast, different manner of bacteria. Whatever was on that pomace was in these particular containers. You can see that during the cold months, that Brett became unrecoverable, couldn't find it. So there was more of a swing up and down in terms of population. But still out in here and out there, you can still detect it. So that wasn't good news in some ways. So can heating the waste destroy Brett? We placed the inoculated pomace in containers. And remember, we had 2 different levels of alcohols, or 2 different levels of sugar, therefore 2 different levels of alcohol. We had one that's low and one that's high. And we use water bath at different temperatures, and we determine culturabilities over time. Pretty straightforward. Here are the data, which are really interesting, because here we have the culturability against the incubation time of heated pumice. So right up here, we're heating pumice at 40 degrees C for different times, 0, 1, 5, or 10 minutes. We then take a sample, we put it into a recovery medium, and we're trying to recover any injured cells, any cells that are still alive. But we'd incubate that for a long period of time, upwards of 60 days, because we wanted to make sure, we wanted to absolutely make sure that we were recovering all viable cells to see which of these processes would actually eliminate Brett. And I think you can see in the low alcohol, we did get viability. Now we had an added treatment here, because here we have the blue is without any SO2 added to those pumice. The red had 45 total, just added. So if we look at the low alcohol, on day 1, you still had viable cells in the without SO2 sample, but that was dropped down, that population significantly decreased by adding some SO2. Now, over time, we started to recover from cells from the red. And you can see there and there. Okay, so 10 minutes at 40 Celsius isn't enough. At 15 degrees C, we see more kill. I'm sorry, at 15 degrees alcohol, we see more kill. So here, with or without SO2, we're not getting any recovery. But if we incubate for a longer period of time, we start to recover cells there and there. So that tells us there was a very low population, but it could recover over time at this temperature. So being microbiologists, let's torture Brett even further. Let's raise the temperature. Let's go up to 50. Here, at 10 minutes, at 50 degrees C, it didn't matter how much alcohol was in that, didn't matter of the residual alcohol in those pumice samples. Didn't matter. Did not see any recovery after 60 days. So I'm very comfortable to say that at that temperature and time, it destroyed Brett. If we half the time to 5 minutes, you can see we only recovered Brett from the samples without SO2. With SO2, nothing, even 60 days. Now, hopefully it's starting to paint a picture for you that this is one of the big tools in your toolbox against Brett. Hurdles. You want to put in place as many hurdles as you possibly can, because there's interactive effects that go on. So here we've got an interactive effect between SO2, temperature, and time. That's how you can defeat it. Let's raise the temperature of the pumice samples. Let's go to 60. 5 minutes was enough at either one of these samples to knock out Brett. Even 1 minute was enough to knock out of the samples that had SO2 present, not the ones that did not have SO2. At 70, no doubt. 1 minute at 70 degrees, you still have no Brett, even after 60 days of incubation. So you can see the temperature time, plus alcohol, plus SO2. All those factors are inhibitory to Brett. And that's one way of control. So the unfortunate side to the work was that Brett can survive in pumice placed in vineyards. There are enough nutrients, there are proper conditions. It can even survive the cold winters. So even though these containers were kind of on the top of the ground, they had a covering on them to keep dust out and such. but we still had got recovery from the Brett. It does depend upon initial populations. So if we're starting with very, very high populations, which over time slowly decrease. So if you have a lower population, odds are good it's going to disappear, but there could be still a few cells in there, depending on how much sample you actually are analyzing. So our recommendation for pumice is to be very careful with its disposal. If you do suspect you've got a Brett infection in the pumice, you're going to have to achieve specific temperatures and time if you're composting material. And this is the problem with composting. The difficulty is that you have a massive temperature gradient going from the center of the pile or the center of the material outwards. And outside can be far, far different in temperature than the inside. So there has to be a constant turning to get all parts of the material exposed to a warm temperature, if not a hot temperature. That's key there. One of the other questions I get a lot of is about filtration. I think I've only met one or two winemakers that actually enjoy, to a certain degree, filtering. Most folks don't enjoy doing it. It's expensive, there's quality concerns. The problem is that VBNC cells, it turns out, could exhibit some form of dwarfing. And what that means is that the size of Brett could change. Now, we've seen a little bit of that, but not consistently. It seems that strains play a role in this. But most definitely, and the biggest concern for winemakers is going to be filtered wine may have a problem depending on the pore size that is selected. So we needed to find, get some research data to help support a recommendation for the industry. Well, what we decided to do is set up an experimental design where we had no SO2 and then 0.5 milligram per liter of molecular SO2. That's a concentration that many wineries will try to achieve during the barrel aging of a wine. And of course, we were worried that SO2 being a stress to the yeast, that could induce VBNC and therefore could induce dwarfing of some sort. We didn't see it. Being good scientists, we always try for 3 replicates. The replicate here, though, is a filter cartridge. And that's why this work was, excuse me, very time consuming. But the grad student did just a wonderful job with it. But we had to change out the filter cartridges, several times in order to get the necessary replication. And there's the treatments that I'm going to be presenting data from. One of the difficulties working with Brett, and it just is what it is such a slow growing rascal that you'll see the horizontal axis on these data. It goes out a long ways, and we have to incubate for such a long period of time, but I think it's worth it. Okay, here's strain B1B. We saw this one before, and we have plotted the filtration replicates themselves as the different symbols. And what we would do is that we would get a nice big population of Brett. We'd hit it with SO2, then a week later we would filter just to see what would happen. And here we're using 1.2 micron absolute. And you can see, as expected, even though we started with a pretty strong population, went down to below detection limits, and it stayed that way for over 212 days. So I'm pretty comfortable saying that Brett was removed from these particular wines. Well, let's not add SO2, and let's just see if 1.2 micron by itself is enough to remove the Brett. So here we did not add SO2, as expected, went to undetectable levels, and wow, sounds good. Greater than 212 days after, we're still not seeing any growth whatsoever. There's been no bloom, no infection, no evidence of any kind of infection. Well, for the sake of argument, And this is part of what I do for a living. Those data are good, but it's always positive to change the organism, just change the strain, because we do know something about Brett. And one of the things we really know is the diversity. It's a really diverse microorganism exhibiting different types of responses to different situations. So here we changed strains to F3, another one which was previously isolated from a Washington wine. Again, we're adding SO2, waiting about a week, and then filtering through 1.2 micron. Perfect. No salt, we're not getting any recovery whatsoever, greater than 250 days after. Perfect, perfect. But if we repeat the previous experiment and not add any SO2 and just put it through a 1.2 micron, well, population drops to undetectable levels there. But look way out here. We've got 2 of the 3 replicates. 2 of the 3 all of a sudden starting to show growth. And that tells you that there were, yeah, it really reduced the number of cells, but did not completely eliminate. So some cells got through this membrane without the SO2 being present and just did not. So this is not good. We then took the same strain and reset the experiment again, but this time we used a 0.8 micron. And you can see that, again, without any SO2 present, running a wine that's very heavily contaminated, huge numbers of cells. Incubating that, we did not see any reoccurrence of the infection 270 days after. So we're very comfortable in our recommendation that a 0.8 micron absolute is the way to go to remove Brett. It will certainly not remove the lactics or acetobacter or any of the bacteria, because that will require a much smaller porosity. Well, one of the arguments that I've heard over the years is you know, if you add way too much nutrient to a grape must to make sure your alcoholic gets to completion, that you run a risk. And the risk is that there would be so much nutrient present in that wine left over from alcoholic that could feed spoilage. So a good question is, if more nitrogen is added to a grape must, is more nitrogen going to be in the resultant wine? And really, does a higher nitrogen encourage infections by this particular yeast? Really, the question that's being asked is, what amount of nitrogen, amino acids, ammonium, etc., what is really needed to support a spoilage event, to support the growth of Brett? So we actually did some experimentation with a different project, but we were producing these synthetic wines, and we'd start with a synthetic grape juice that really mimicked, as best we could, the various nutrients that would be present in a Cabernet Sauvignon grape must. So this is completely synthetic. And we set it up so that we had 2 different levels of nitrogen in the must, 150 and 250, and then 3 different levels of sugar. And we just fermented to dryness. Or fermented, yeah, I think most of these went dry, if I remember right. We looked at the wine, and as you might imagine, the must, when you have a higher level of nitrogen in the must, yes, you do get a higher level of alpha amino nitrogen and also of ammonia nitrogen. Okay, that makes sense. This is a different yeast strain. So we changed the yeast here, same experimental design, 2 levels of yan, and then 3 levels of sugar. And yes, yet again, you're seeing more residual nitrogen in the wine and more ammonium. Okay. For simplicity of the slides, I'm going to say if the must had 230 grams per liter of sugar, that's how much ethanol yield. But this will be quote unquote the lower sugar starting point. And there's the 250 gram per liter of sugar. And then there's the 270. Okay. Well, we took those wines that came from the two different yeast, and we thought, it'd be interesting to see if Brett can grow in that, just to see what would happen. And is there a difference between how Brett responds given how much nitrogen was originally present in the grape must? So here are the low ethanol. So this would be the lower amount of sugar initially in the grape must, which yields a lower amount of ethanol. But we have low yan versus high yan. So the 150 versus 250 in the grape must. I don't see much difference there. Let's go to the medium amount of ethanol. Well, Brett still grows. There's a little bit of a delay, but I'm not seeing any difference between the low yan must or the high yan must. If we go to the high ethanol, still no real differences between the two musts, low versus high. So there seems to be more of an influence of the original sugar and therefore the amount of ethanol that's produced, as opposed to anything to do with nitrogen. Nitrogen does not seem to affect Brettanomyces, at least in this experimentation. So how much nitrogen is actually needed by Brett? How much does it need to grow? We made another synthetic wine, and this time we started with the wine. We didn't ferment it. It had 13% alcohol in it, had various nutrients in it, but yan was limiting. And so we carefully added back yan. So here we have 0, and then we have different levels. We thought for sure that growth would probably be around in this area someplace. But there's the 0. So it didn't grow a little bit maybe, but it just, we could recover it. It didn't die off, but it just is there. But here are the data. Even with 6 milligrams nitrogen per liter, 6, you're still seeing a 2 log increase in population. That's spoilage. That's a spoilage event. So the translation is that Brett does not need a lot of nitrogen to do its thing. Sorry. One of the questions I've gotten in recent times has been about chitosan. What is it? It is a derivative of chitin. So they actually harvest chitin from different sources, shellfish, fungi, things like that, mushrooms. It's it serves as a structural biopolymer. So it'd be similar in function to cellulose in plants. And this is what it actually is. Here on the top you have chitin, which is in its natural form, and chitosan, which is a different type of a molecule. And where you look is right here. You can see you've got a 2-carbon group that has been added to that nitrogen right there that's absent there. Now, normally for microbiological purposes, you're dealing with chitosan that has less than 25% of these nitrogens have this acetyl group on them. That's why they give it a different name. But that's also part of the purchasing, because all the different preparations that are out there are going to vary in the degree of acetylation, how much of the polymer has these groups on it. Now, I thought about how to present some of these data because I was only given 50 minutes today. And I thought, sometimes the pictures are worth 1,000 words. Here we've got a population of Brett. It is in media, it's not in wine. But we've had one group that was undisturbed, meaning we added the chitosan and we let it sit. The other one was continuously shaken with the chitosan. I can't remember which actual chitosan product we used. But here we have just samples that were removed and put onto a spread plate. So here we have 0 amounts of chitosan. You can see all that white stuff on there. That is Brett growing like crazy. Huge populations of it. But you can see the undisturbed. If we start taking samples off that contain 2 grams, 4 grams, and higher, you can see a decrease in that population going from plate to plate. So somehow Brett is not being, we're seeing populations decrease to some level, but we're not seeing that same decrease at those same concentrations in the shaken samples. It's not until you get up to this concentration that you don't see any Brett colonies there. You see a few here and a couple there. Well, what's going on here is that there's actually 2 mechanisms by which chitosan works. One is a settling effect, that it'll actually entrap the yeast and force it to the bottom of the liquid, like in that picture. So here we have an SEM, here's chitosan, and here's Brett, kind of been encased, if you will, in the chitosan. But there's also an inhibitory function here as well, that the chitosan itself seems to inhibit the Brett, and away you go. Is it effective? Yes, but you have to be careful if you're using it, because this is going to be a surface phenomena. If you have Brett in the barrel itself that you're adding the chitosan to, you're not going to get rid of the Brett from that barrel. And we'll show you that work in a second. In fact, let's show it right now. I'm excited about this. This has been good, wonderful research. I'm proud of the student who was part of this. All right, survival in oak staves. This is one of the key things about anybody attempting to use chemicals, and that could be ozone, it could be gassing with SO2, it could be a number of things. Chemicals only penetrate a few millimeters. And Brett has been long suspected of being able to penetrate far deeper into oak than what chemicals can do. And that's why it's so difficult to sterilize wood. If we all had autoclaves that are as big as a 55-gallon barrel, yeah, you can get rid of Brett that way, but not by just adding chemicals. There are factors that affect the yeast penetration we'll talk about today. Oak species, toasting. Where is the stave? Is it in the bottom of the barrel or is it in the top of the barrel? I'll be presenting work involving steam primarily, but hot water can also be used as well. Heat does destroy microbes like Brett, and that's the good news. Inactivation begins at around 50 Celsius, but there's a relationship there that we showed earlier on in the work. There's a relationship between temperature and time. Lower temperature, longer time. There's something called a D value, and it's the time at a given temperature for a log reduction. At 55 Celsius, if you read through the literature, it's somewhere between 12 to 80 seconds for 1 log reduction. So again, it points to, yes, you can help limit Brett by using temperature, but there's a relationship between it and time that is extremely important. There's also the factor of initial population. Higher populations take more temperature and more time. So that's just that's just a general a microbiological concept, if you will. Very, very important. Okay, so what we did, we took barrels, we added wine, we infected the wine with Brett, and we let them sit, I think, 3 months, and then we took them apart. We took a table saw and we cut the staves into blocks. We then use a band saw to saw cross sections, which were 4 millimeters thick. And if you've never played with the bandsaw, they're great. They're great until the blade breaks, and then you have all sorts of fun. But they are really an incredible tool. We then would take those pieces, those individual sections, if you will, of the stave, and we incubated them a very long period of time in a recovery medium, up to 90 days. The problem with a lot of the past work that has been done is that their sample size was so small that if they said, well, the yeast was dead, we didn't recover any yeast from that very small sample size, you still may have a very, very, very low population that over time could infect that wine, future wine, in that barrel. So we were looking at a situation where we wanted to have a large surface area. And so here's what we actually did. Here's a piece of that 3x10, that chunk of the stave. Notice the little marks that are made here. Those are every 4 millimeters. I would then put them on my bandsaw. And yeah, that's a beautiful picture of my hands. And don't worry, I still have all my fingers. So we didn't have any accidents. But those cross sections look like that. And so we took this entire cross section and put that into recovery medium, or sometimes we'd use a low alcohol wine, just to see if we can grow Brett again. So I'm really comfortable with these data that we're going to present to you today, because our detection limit is really, really, really low. Here's a student filling the barrels. We used a smaller barrel because we want to take them all apart. So we use 16-liter barrels, American and French oak, both light and heavy toast. And the questions we had is, where in the barrel would we find the yeast? The second was about heating, because heat is commonly used as a means to hopefully reduce populations in those barrels. Okay, here are the top staves of those 16-liter barrels. Here we have culturability. And what this basically represents is that we took those cross striations, put them into the recovery medium, and incubated that. So all of these data here are all about what is in either the recovery medium or the wine that we put in these stave pieces in. French versus the American. First off, you can see that we are getting, Brett, commonly higher populations in the French compared to the American. They were all in that first piece, that first section of a cross section of the stave. So the 0, which is the closest to the wine, to the 4 millimeters, that's where we're seeing the Brett penetrate. But all had populations in them. These are the top staves. If we go to the bottom staves, these would be ones that have much more contact with the wine. The top staves don't. There's some contact, there's some wicking that's going to go on, but the bottom staves constantly in contact. So here again, the French oak had a slightly higher population than the American oak. They all reached very high populations in that recovery medium. Now that's at the 0 to 4 piece. But if you look over here, here you've got French, the heavy toast, versus French, the light toast. And after incubation of at least a couple of weeks, if not almost 3 weeks, we started to see some populations at this depth, the 5 to 9 millimeter. So these data indicate to us that the yeast was able to penetrate further into the French oak, in particular, the heavy toast. Certainly heavy toast can have more cracks, crevices, and areas where the yeast can penetrate. These data support that. We did not find the yeast past 5 millimeters in the American. It just would not penetrate as easily. Okay, so we got an idea of the effect of type of oak and the level of toasting. What about heating? We took those same staves, those little blocks, and we put them on a custom-made stainless steel plate that looked like that. We then had all these thermocouples right here going at different levels within those staves. And that's a close-up of all the pieces. We then put it over a steam kettle and recorded temperature over time. Periodically, we would remove the blocks and determine yeast recoveries. This is cool. This is cool. Okay. These are a little complicated, but let me walk you through. Here we have culturability, so population. This would be the American oak 16-liter barrels, light toast, American heavy toast. Here we have the 0 to 4 millimeter layer, so the one that's closest to the wine. Here we have the 5 to 9 millimeter layer, farthest from the wine. And here we have time. So this is incubation time. Here, 12 minutes, either barrel, we saw destruction, no recovery whatsoever. Even 9 minutes, we're seeing no recovery. At 6 minutes, it took 30 days to start to see some recovery, and that was only from the 0 to 4 millimeter. In fact, if you go to the 0, we don't see any whatsoever. We don't see any recovery whatsoever. Oop. Here's the French. Here's the French light toast and the French heavy toast. Here, the yeast could be found in that 5 to 9 millimeter. Now, 12 minutes of steaming saw no recovery. But interesting, it's that 5 to 9 millimeter that you start to see some recovery of the yeast. And that's after 9 minutes of heating. So if the yeast is that far down in the barrel, past 5 millimeters in this one particular section, it's going to take you longer to get rid of it. Here you saw that the removal of that yeast. So what can we conclude? Brett does penetrate oak staves approximately 8 to 10 millimeters. It is affected by species and toasting, and it's going to be difficult to reach using chemicals. You're gonna, if you have a Brett barrel, you're going to have to steam for some significant time to get rid of it. Unfortunately, you're not going to know the depth of penetration for any given barrel, so there's going to be some guesswork here. Give you an idea of hot water. You get equivalent results on that 0 to 4 millimeter cross section. 70 Celsius takes 20 minutes. At a higher temperature of water, 80 degrees, only 15. In the up to 9 millimeters penetration in that stave, it's going to take longer. 70 Celsius for 30, 80 Celsius takes 20 minutes. We also took apart some commercially infected barrels, and we used a Forstner bit. I don't know if you've ever used one, but there it is right there. They will cut a flat bottom in a hole. So you can get an app, so you can really dial it in to figure out where the yeast is actually located using one of these. So we removed 2 millimeter increments. We incubated all these shavings in the media. And these are the data. Now, these are commercial barrels now. Here we have a 225 liter American medium toast. And here's the yeast penetration. Here's a French medium, low, and toast level. Notice it penetrates farther. That's what we've seen in the past. But look at these. In that second section, 2 millimeters and above, see the populations go up? That made absolutely no sense to us whatsoever. And we could not figure out what was going on until we talked to the winery. They've been SO2 gassed. So they got a reduction in population at that right at the interface with the wine, but they had a higher population deeper in that barrel because the chemicals could not reach. So how do we really control Brett and really other spoilage microbes? You got to put in hurdles, and hurdles are anything that's slow spoilage. It could be SO2, temperature, temperature of the cellar, alcohol level of the wine, anything that you can put in place. The trick is, if you have too few hurdles, you're going to have an increased risk of spoilage, but too many, it could be the expense is higher than the benefit. And that's where winemakers need to get that balance. But as we've demonstrated, and this is the key part here, folks, understand that there are interactions between all these different variables. Those will help in terms of limiting spoilage. I found this in my slide selection, and this was a stone that is put in, that is present in Napa, in downtown Napa. And I decided to use this because he, again, a wonderful, wonderful, wonderful quote. Vigilance and discipline are the winemaker's best friends. Have the vigilance to keep looking for problems, at the same time, the discipline to deal with them. And I want to thank everybody for 32 years of wonderful support. You folks have just been phenomenal. And so thank you very much.
Great. So if you have any questions, go ahead and put them in the Q&A. We've got a few minutes, about 5-7 minutes before it's 4 o'clock, before we turn into pumpkins. And if no questions come to mind right now, you can email me, you can email Charlie with if something comes up down the road and you, or before you start fresh, if something comes to mind and you want to ask him.
I'll add one more piece of information that's come up very recently. I've been reading up on some interesting work that's being done in Europe. They claim that there are some lactic acid bacteria out there that are capable of producing 4-ethylphenol and other volatile phenols. Now, this had long been rumored, but a lot of people had said, no, it's only Brettanomyces. So if you have a high concentration of those metabolites, then it's definitely Brett. Well, there's at least some information now to point to one of the lactics that is present in Washington wines, and that's plantarum, Lactobacillus plantarum, which is sometimes used for malolactic. Now, there's still some questions on the research, and I'm still not completely sure about it. But that may be something just to put into the back of your mind as well.
Okay, we've got one. Charlie, why is racking after 10 days necessary when using chitosan?
Because you're trying to leave it behind. Because remember, you have that mechanism by which the chitosan seems to surround the Brett and bring it out of the wine itself and keep it in the bottom of the barrel. And that's exactly what it's doing. There's been a number of studies that have shown that particular phenomena. So I'm pretty comfortable with that one.
Okay. Any others?
Folks, you know where I am. And as Melissa said, you can always shoot me an email or, shoot me a question. I am so, in the last minute, if there aren't any other questions, one thing I do want to say, and I'm just going to say it because I'm really old. This industry has grown so much when it comes to Brett, because I can remember when you ask a winemaker about it, there was, folks got defensive and they didn't want to talk about it. And it was clearly, if a wine got infected with Brett, it was clearly the winemaker's fault, and people were fired because of it. But we have really grown and with our understanding of what Brett is and what it can do and how it can survive. And that's through you folks. And I'm really very, very grateful for all the support we've got, we have received. And, thank you a lot, because now we can talk about it. Now we don't have to hide it under the rug.
Right. Okay, great. Well, expect to see a recording of the link here in a couple of days, and I will sign off, and let's cross our fingers for a wonderful vintage and harvest and crush in the next coming month or so.
Absolutely. Thanks, everybody.
Good luck, everyone. Oh, Charlie, do you see there's a message there in the chat for you?
It's making me cry.
Okay.
Folks, you know, I've been doing this 32 years and you know, it's still fun. It's still exciting. We were doing research this morning on the blackboard. It's still a lot of fun. And God bless everybody. Stay safe, folks, whatever you do. And I agree with Melissa. Have a great harvest.
Okay, thanks.